So hello dear learners, in this video we are going to study chapter 13 plant growth and development, and in this chapter we will cover some topics that growth, differentiation, redifferentiation and redifferentiation. Third, development, and fourth, plant growth regulators. So here we will start our topics. So you have already studied that the organization of flowering plants in chapter 5, and so read with us because reading is the first steps towards a good learning. Have you ever thought about where and how the structures like root systems, leaves, flowers, fruits, and seeds arise, and that too in an orderly sequence? You are by now aware of the term seed, seedling, plantlet, mature plant. You have also seen that trees continue to increase in height or girth over a period of time. However, the leaves, flowers, and fruits of the same tree not only have limited dimensions but also appear and fall periodically and sometimes repeatedly. Why does the vegetative phase precede flowering in plant? All plant organs are made up of variety of tissues. Is there any relationship between the structure of a cell, a tissue, an organ, and the function they perform? And the structure and the function of these be altered. All cells of the plants are descendants of the zygote. The question is then why and how do they have different structural and functional attribute. Development is the sum of two processes, growth and differentiation. To begin with, it is essential and sufficient to know that the development of a mature plant from a zygote fertilized egg follow a precise and highly ordered succession of events. During this process, a complex body organization is formed that produces roots, leaves, branches, flowers, fruits, and seeds, and eventually they die. The first step in the process of plant growth is seed germination. The seed germinates when favorable conditions for growth exist in the environment. In absence of such favorable conditions, the seed do not germinate and goes into a period of suspend growth or rest. Once favorable conditions return, the seeds resume metabolic activities and growth takes place. In this chapter, you shall also study some of the factors which govern and control these developmental process. These factors are both intrinsic, internal, and extrinsic, means external to the plant. Coming to the next topic, that is growth. So growth is regarded as one of the most fundamental and conspicuous characteristics of a living being. What is growth? Growth can be defined as an irreversible permanent increase in size of an organ or its part of or even of an individual cell. Generally, growth is accompanied by metabolic processes, both anabolic and catabolic, that occur at the expense of energy. Therefore, for example, expansion of a leaf is growth. How would you describe the swelling of a piece of wood when placed in water? Next is plant growth generally is indeterminate. Plant growth generally is indeterminate. Plant growth is unique because plants retain the capacity for unlimited growth throughout their life. This ability of plants is due to the presence of meristems at certain locations in their body. The cells of such meristems have the capacity to divide and self-perpetuate. The product, however, soon loses the capacity to divide, and such cells make up the plant body. This form of growth, wherein new cells are always being added to the plant body by the activity of meristem, is called the open form of growth. What would happen if the meristem ceases to divide? Does this ever happen? In earlier classes, you have studied about the root apical meristem and shoot apical meristem. You know that they are responsible for the primary growth of the plants and principally contribute to the elongation of plants along their axis. You also know that in dicotyledons, the plant and gymnosperm, the lateral meristems, vascular cambium, and cork cambium appear later in life. These are meristems that cause an increase in the girth of organs in which they are active. This is known as secondary growth of plant. Next is growth is measurable. Growth is measurable. Growth at a cellular level is principally a consequence of increase in amount of protoplasm. Since increase in protoplasm is difficult to measure directly, one generally measures some quantity which is more or less proportional to it. Growth is therefore measured by a variety of parameters, some of which are increase in fresh weight, dry weight, length, area, volume, and cell number. You may find it amazing to know that one single maize root apical veg stem can give rise to more than 17,500 new cells per hour, whereas cells in a watermelon may increase in size by up to 350,000 times. In the former, the growth is expressed as increase in cell number. The latter expresses growth as increase in size of the cell. While the growth of pollen tube is measured in terms of its length, an increase in surface area denotes the growth in the dorsiventral leaf. Next is phases of growth. The period of growth is generally divided into three phases, namely meristematic, elongation, and maturation. Let us understand this by looking at the root tips. The constantly dividing cells both at root apex and the shoot apex represent the meristematic phase of growth. The cells in this region are rich in protoplasm, possess large conspicuous nuclei. Their cell walls are primary in nature, thin and cellulosic with abundant plasmodesmatal connections. The cells proximal, just next away from the tip, to the meristematic zone represent the phase of elongation. Increased vacuolation, cell enlargement, and new cell wall deposition are the characteristics of the cells in the sheath. Further away from apex, more proximal to the phase of elongation, lies the portion of axis which is undergoing the phase of maturation. The cells of this zone attain their maximal size in terms of wall thickening and protoplasmic modifications. Most of the tissues and cell types you have studied in earlier classes represent this phase. Next is growth rates. The increased growth per unit time is termed as growth rate. Thus, rate of growth can be expressed mathematically. An organism or a part of organism that can reproduce more cells in a variety of ways, like arithmetic, geometric, zygote division. So here is the diagrammatic representation of arithmetic, geometric growth and stages during embryo development showing geometric and arithmetic phases. The growth rate shows an increase that may be arithmetic or geometrical. In arithmetic growth following mitotic cell division, only one daughter cell continues to divide while the other differentiates and matures. The simplest expression of arithmetic growth is exemplified by a root elongating at a constant rate. Look at the figure 13.5. When plotting the length of the organ against time, a linear curve is obtained. Mathematically, it is expressed as LT equal L0 plus RT. LT equal length at time t, L0 equal length at time 0, and R growth rate or elongation per unit time. Let us now see what happens in geometrical growth. In most systems, initial growth is slow and it increases rapidly thereafter and exponential rate. Here, the both are progeny cells followed by mitotic cell division, retain the ability to divide and continue to do so. However, with limited nutrient supply, the growth slows down, leading to the stationary phase. If we plot the parameter of growth against time, we get a typical sigmoid or S curve. A sigmoid curve is a characteristic of living organism growing in a natural environment. It is typical for all cells, tissues, and organs of the plant. Can you think of more similar examples? What kind of curve can you expect in a tree showing seasonal activities? The exponential growth can be expressed as V1 equal V0 ERT, V1 equal final size, weight, height, number, etc. V0 equal initial size at the beginning of the period, R equal growth rate, T is the time of growth, E is the base of natural logarithms. Here is the relative growth rate and is also the measure of ability of the plant to produce new plant material referred to as efficiency index, and the final size of V1 depends on the initial size V0. Quantitative comparison between the growth of living systems can also be made in two ways. First, measurement and the comparison of total growth per unit time is called the absolute growth rate. Second, the growth of given system per unit time expressed on a common basis. Example, per unit initial parameter is called the relative growth rate. In figure 13.7, two leaves A and B are drawn that are of different sizes but shows absolute increase in area in the given time to give leaves A1 and B1. However, one of them shows much higher relative growth rate, H1 and Y. Next, conditions for growth. Next is conditions for growth. Why do you not try to write down what you think are necessary conditions for growth? This list maybe have water, oxygen, and nutrients as well as initial elements for growth. The plant cell grow in size by cell enlargement, which in turn requires water. Rigidity of cells helps in extension of growth. Thus, plant growth and further development is intimately linked to the water status of the plant. Water also provides the medium for enzymatic activities needed for growth. Oxygen helps us in releasing metabolic energy essential for growth activities. Nutrients, macro and micro essential elements are required by plants for synthesis of protoplasm and act as a source of energy. In addition, every plant organism has an optimum temperature range best suited for its growth, and a deviation from this range could be detrimental to its survival. Environmental signals such as light and gravity also affect certain phases or stages of growth. Next is differentiation, dedifferentiation, and redifferentiation. The cells derived from root apical and shoot apical meristems are cambium differentiate and mature to form specific function. This act leading to maturation and stem differentiation. During differentiation, cells undergo few to major structural changes both in their cell walls and protoplasm. For example, to form a tertiary element, the cells would lose their protoplasm. They also develop a very strong elastic lignocellulosic secondary cell walls to carry water to long distances, even under extreme tension, to try to correlate the various anatomical features you encounter in plants to function they perform. Plants show another interesting phenomena. The living differentiated cells that by now have lost the capacity to divide can regain the capacity of division under certain conditions. The phenomena is termed as redifferentiation. For example, formation of meristems, interfascicular cambium and core cambium from fully differentiated parenchyma cells. However, while doing so, such meristems or tissues are able to divide and reproduce cells that once again lose the capacity to divide but mature to perform specific function. It means get redifferentiated. List some of the tissues in a woody dicotyledon plant that are products of redifferentiation. How would you describe a tumor? What would you call the parenchyma cells that are made to divide under the controlled laboratory conditions during plant tissue culture? Recall in section 13.1.1 we have mentioned that the growth in plants is open. It means it can be indeterminate or determinate. Now we may say that even differentiation in plants is open because cells or tissues arising out of the same meristem have different structures at maturity. The final structure at maturity of cell or tissue is also determined by the location of the cell within. For example, cell positioned away from the root apical meristem differentiate as root capsules, while those pushed to the periphery mature as epidermis. Can you add a few more examples? Open differentiation correlating the position of a cell to its position in an organ. Next is development. Development is a term that includes all changes that an organism goes through during its life cycle, from germination to a seed to senescence. Diagrammatic representation of the sequence of processes which constitute the development of a cell is of a higher plant is given in figure 13.8. It is also applicable to tissues or organs. Plants follow different pathways in response to environment or phases of life to form different kinds of structure. This ability is called plasticity. Example: heterophyly in cotton, coriander, and larkspur. In such plants, the leaves of juvenile plant are different in shape from those in mature plants. On the other hand, difference in shapes of leaves produced in air and those produced in water in buttercup also represent the heterophilous development due to the environment. This phenomenon of heterophyly is an example of plasticity. Thus, growth, differentiation, and development are very closely related events in life of a plant. Broadly, development is considered as the sum of growth and differentiation. Development in plants is under control of intrinsic and extrinsic factors. The former include both intracellular, genetic, or intercellular factors, chemicals such as plant growth regulators, while the latter includes light, temperature, water, oxygen, nutrition, etc. Next is plant growth regulators. Plant growth regulators. Characteristics. The plant growth regulator PGRs are small, simple molecules of diverse chemical composition. They could be indole compounds, indole 3-acetic acid, IAA, entenine derivatives, N6, furfurylaminopurine kinetin, derivations of carotenoids, abscisic acid, ABA, terpenes, gibberellic acid, GA3, or gases, ethylene C2H4. Plant growth regulators are variously described as plant growth substances, plant hormones, or phytohormones in literature. In PGRs can be broadly divided into two groups based on their functions in a living plant body. On a group of PGRs is involved in growth promoting activities such as cell division, cell enlargement, pattern formation, tropic growth, flowering, fruiting, and seed formation. These are also called plant growth promoters. Example: auxins, gibberellins, and cytokinins. The PGRs of other group play an important role in plant responses to wounds and stresses of biotic and abiotic origin. They are also involved in various growth inhibiting activities such as dormancy and abscission. The PGR abscisic acid belongs to this group. The gaseous PGR ethylene could fit either of the groups, but it is largely an inhibitor of growth activities. Next is the discovery of plant growth regulators. Interestingly, the discovery of each of the 5 major groups of PGRs have been accidental, and all this started with observation of Charles Darwin and his son Francis Darwin when they observed that the coleoptiles of canary grass responded to an unilateral illumination by growing towards the light source, phototropism. After a series of experiments, it was concluded that the tip of coleoptile was the site of transmittable influence that caused the mending of the entire coleoptile. Auxin was isolated by F.W. went from tips of coleoptiles of oat seedlings. The Bacni foolish seedling disease of rice seedling was caused by a fungal pathogen Gibberella fuzzicuroi E. Kurosawa. Reported the appearance of symptoms of the disease in rice seedling when they were treated with sterile filtrates of fungus. The active substances were later identified as gibberellic acid. F. Skoog and his co-workers observed that from the internodal segments of tobacco stems, a calasamas undifferentiated cells proliferated on leaf. In addition to auxins, the nutrient medium was supplemented with one of the following extracts of vascular tissues: yeast extract, coconut milk, or DNA. Miller et al. 1955 later identified and crystallized in cytokinesis promoting active substances that they termed kinetin. During mid-1960s, the 3 independent researchers reported that purification and chemical characterization of 3 different kinds of inhibitors, inhibitor B, abscission, second on domin, later all 3 were proved to be chemically identical. It was named abscisic acid ABA. HH cousins in confirmed the release of volatile substance from ripened oranges that hastened the ripening of stored unripened bananas. Later, this volatile substance was identified as ethylene, a gaseous PGR. Let us study some of the physiological effects of these 5 categories of PGRs in next section. Next is physiological effect of plant growth and regulators. Auxins. Auxins from Greek auxin to grow was the first isolated from human urine. The term auxin is applied to the indole 3-acetic acid IAA and to other unnatural and synthetic compounds having certain growth regulating properties. They are generally produced by growing apices of the stems and roots, from where they migrate to the regions of their action. Auxin like IAA and indole bitric acid IBA have been isolated from plants. NAA and naphthalene acetic acid to 4-dichlorophenoxyacetic acid and synthetic auxins. All these auxins have been used extensively in agricultural and horticultural practices. They help to initiate rooting in system cuttings and application widely used for plant propagation. Auxins promote flowering, example in pineapples. They help to prevent fruit and leaf drop at early stage but promote the abscission of old mature leaves and fruits. In most higher plants, the growing apical bud inhibits the growth of lateral auxiliary buds, a phenomena called apical dominance. The removal of shoot tips decapitation usually results in the growth of lateral buds. It is widely applied to tea plantations and hedge making. Can you explain why? So answer this question. Auxin also induce parthenocarpy, example tomatoes. They are widely used as herbicides 2,4-D, 2,4-dichlorophenoxyacetic acid, widely used to kill dicotyledons. Weeds does not affect the mature monocotyledons plants. It is used to prepare weed-free lawns by gardeners. Auxin also controls xylem differentiation and helps in cell division. Gibberellins. Gibberellins are another kind of promotory PGR. There are more than 100 gibberellins reported from widely different organisms such as fungi and high plants. They are denoted as GA1, GA2 and GA3 and so on. However, gibberellic acid was one of the first gibberellins to be discovered and remains the most intensively studied form. All GAs are acidic. They produce a wide range of physiological responses in plants. Their ability to cause an increase in length of axis is to increase the length of grapes stalks. Gibberellins cause fruit like apple to elongate and improve its shape. They also delay senescence. Thus, the fruits can be left on tree longer so as to extend the market period. GA3 is used to speed up the malting process and weaving industry. Sugarcane stores carbohydrate as sugar in their stems. Spraying sugarcane crop with gibberellins increases in the length of a stem, thus increasing the yield by as much as 20 tons per acre. Spraying juvenile conifers as GAs hastens the maturity period, thus leading to early seed production. Gibberellins also promote bolting, internode elongation just prior to flowering in beet, cabbages, and many plants with roset habit. Next are cytokinins. Cytokinins have a specific effects on cytokinesis and were discovered as the kinetin, a modified form of adenine and purine from autoclaved herring sperm DNA. Kinetin does not occur naturally in plants. As for natural substances with cytokinin-like activities led to isolation of chitin from corn kernels and coconut milk. Since the discovery of chitin, several naturally occurring cytokinins and some synthetic compounds with cell division promoting activity have been identified. Natural cytokinins are synthesized in regions where rapidly cell division occurs. For example, root apices, developing shoot buds, young fruit, etc. It helps to produce new leaves, chloroplast in leaves, lateral shoot growth, and adventitious shoot formation. Cytokinins help overcome the apical dominance. They promote nutrient mobilization, which helps in delay of leaf senescence. Ethylene. Ethylene is a simple gaseous PGR. It is synthesized in large amounts by tissues undergoing senescence and ripening fruits. Influences of ethylene on plants include horizontal growth of seedlings, swelling of the axis, and apical hook formation in dicot seedling. Ethylene promotes senescence and appreciation of plant organs, especially of leaves and flowers. Ethylene is highly effective in fruit ripening. It enhances the respiration rate during ripening of fruits. This rise in rate respiration is called respiratory climactic. Ethylene breaks seed and bud dormancy. Initiates germination in peanut seeds. Sprouting of potato tubers. Ethylene promotes rapid internode petiole elongation in deep water rice plants. It helps leaves or upper parts of the shoot to remain above water. Ethylene also promotes root growth and root hair formation, thus helping the plants to increase their absorption surface. Ethylene is used to initiate flowering and for synchronizing fruit set in pineapples. It also induces flowering in mango. Since ethylene regulates so many physiological processes, it is one of the most widely used PGR in agriculture. The most widely used compound as source of ethylene is ethifone. Ethifone in an aqueous solution is readily absorbed and transported within the plant and releases ethylene slowly. The ethifone hastens fruit ripening in tomatoes and an apple and accelerates absorption in flowers and fruits. Thinning of cotton, cherry, walnut. It promotes female flowers and cucumbers, thereby increasing the yield. Abscisic acid. As mentioned earlier, abscisic ABA was discovered for its role in regulating abscission and dormancy, but like other PGRs, it also has other wide-ranging effects on plant growth and development. It acts as a general plant growth inhibitor and an inhibitor of plant metabolism. ABA inhibits seed germination. ABA stimulates the closure of stomata and increases the tolerance of plants to various kinds of stresses. Therefore, it also called the stress hormone. ABA plays an important role in seed development, maturation, and dormancy. By inducing dormancy, ABA helps seed to withstand desiccation and other factors unfavorable for growth. In most situations, ABA act as an antagonist to GAS. We may summarize that for any and even phase of growth, differentiation, and development of plants, one or the other PGR has some role to play. Such roles could be complementary or antagonistic. These could be individualistic or synergistic. Similarly, there are a number of events in the life of plant where more than one PGR interact to affect that event. Example: dormancy in seeds or buds, abscission, senescence, and apical dormance, etc. Remember, the role of PGR is only if one kind of intrinsic control along with genomic control and extrinsic factors. They play an important role in plant growth and development. Many of the extrinsic factors such as temperature and light control plant growth and development via PGR. Some of such events could be vernalization, flowering, dormancy, seed germination, plant movements, etc. We shall discuss briefly the role of light and temperature, both of them the extrinsic factors on initiation of flowering. Next is summary, and chapter is almost finished. So let us study the last topic, the summary of the chapter. Growth is one of the most conspicuous events in any living organism. It is an irreversible increase expressed in parameters such as size, area, length, height, volume, cell number, etc. It conspicuously involved in increased protoplasmic material. In plants, meristems are the sites of growth. Root and shoot apical meristems, sometimes along with intercalaring meristem, contribute to the elongation of growth of plant axis. Growth is determinate and higher plants following cell division and root and shoot apical meristem cells. The growth could be arithmetic or geometrical. Growth may not be and generally is not sustained at a higher rate throughout the life of a cell or tissue or organ or organism. One can define three principal phases of growth: the lag, the log, and the senescence phase. When a cell loses the capacity to divide, it leads to differentiation. Differentiation results in development of structures that is commensurate with the function the cells finally has to perform. General principle for differentiation for cell, tissues, and organs are similar. A differentiated cell may de-differentiate and then re-differentiate. Since differentiation in plants is open, the development could also be flexible. It means the development is the sum of growth and differentiation. Plants exhibit plasticity in development. Plant growth and development are under the control of both intrinsic and extrinsic factors. Intercellular intrinsic factors are the chemical substances called plant growth regulators, PGR. There are diverse groups of PGR in plants, principally belonging to 5 groups: auxin, gibberellin, cytokinin, abscisic acid, and ethylene. These PGRs are synthesized in various parts of the plant. They control different differentiation and developmental events. Any PGR has diverse physiological effects on plants. Diverse PGRs also manifest similar effects. PGRs may act synergistically and antagonistically. Plant growth and development is also affected by light, temperature, nutrition, oxygen status, gravity, and such external factors. So the chapter is finished. So